Thank you, Richard and Harris, for an exciting morning, and our speakers as well. And what I'm going to try to do, I want to thank the ILSI organizers for bringing me back to an ILSI annual meeting. Haven't been here in a while. And I want to, this is a cover slide for our panel, where you've heard a lot about risk and benefit of, or potential risk and potential benefits of caffeine today. And what we want to do is to see if the panel can help us, and perhaps some of you in the audience as well, can help us to see if we can do some balancing of risks and benefits related to the kinds of things you've heard this morning. I've been involved in, my disclosure statement, been involved in caffeine and coffee-related issues going way, way back to 1981. And when I was working at General Foods and Kraft General Foods, and I've since consulted for numerous trade associations and companies and ILSI, INIFIC, and others on caffeine and health issues. This long history, I'll declare, it makes me a friend of caffeine. We ask the question, friend or foe, and everyone knows that I'm a friend of caffeine. What I want to do, I want to give you some framework, and there's some people in the audience who've been very involved in this BRAFO framework. We've got a couple of slides here talking about BRAFO. Benefit risk analysis of foods. It was undertaken by ILSI Europe, funding by the European Commission, and it ran, I think, for about 4 years, and perhaps there's more still to come. Started in 2007, and this is the cover. This is the website, the BRAFO website that you can pull up, and some great links to background papers. An executive summary, by the way, there was an issue of Food and Chemical Toxicology in January 2012 that published 10 papers related to this effort. This one here is a summary paper of the entire project, and Dr. Vidri is next door, so he won't be joining us today. I wish we still had him, but the last author there, Dr. Ciardini, is in the audience, and he's promised to help out if we could use some help on the BRAFO process. This paper was published. This was the consensus working group that met, and this paper was published in Food and Chemical Tox also in 2013. And there's a couple of schematics and frameworks that I want to show you that they put a lot of effort into related to developing this BRAFO project. It's a tiered approach. You can see the tiers going from tier 1 to tier 4. But even before you get into the tier 1, you kind of decide what the problem formulation is, and that's we're going to do today. We're going to try to do that and discuss some of the tier 1 and perhaps tier 2 issues related to caffeine. It's about balancing the benefits and risk. If in the analysis you see no benefit or you see no risk, the project is over. You don't go further. But in tier 2, if you looked at all the individual benefits and risks of the compound or the food that you're working with, you can qualitatively decide whether the risks are outweighing the benefits or the benefits are outweighing the risk, and you can take that further into various quantitative measures in tier 3 and tier 4 to see if you can actually do a quantitation of the risks. Another schematic of this, of the 4 tiers, and you can see on the left side is in red. That's the risk side, starting with hazard identification, all the way down to determining the risk of that individual food or chemical in food. And on the right side, in green, appropriately, the positive health effects. And if you look at these boxes, as I looked at these boxes, I mean, these boxes have been the purview of ILSI for decades. These are the kinds of things that we've put a major effort into. So I'm really proud of the effort that went on in Europe. Europe, by the way, is way ahead of us, and it wasn't just this BRAFO effort. The European Food Safety Authority convened a panel, and they provided guidance in 2010. And this document is a 40-page document available on the EFSA website that describes some similar activities related to what went on in the BRAFO effort. I noticed in digging back through 2 years ago at ILSI annual meeting in Miami, you had a balancing risk and benefits of foods half-day session. And Dr. Richard Williams from George Mason ended his benefit risk presentation by providing this slide. Here's I don't know if that's a high school student or a college student, but when I saw this slide, it reminded me of our beloved Peter Dews, who we no longer have with us. Certainly a great friend of ILSI and a friend of caffeine. And I've had conversations, and several of you have had conversations with Peter. And I can see Peter's face if I were to ask him, what about the benefits and the risk, Peter? I can see Peter's face, and I just love the man. This is hard, Jim. This is hard. So that's a preview of, it's not going to be easy, but let's jump into it. We're going to first have the panel get some panel ideas on the balancing of the risks and the benefits. And after that, I'd like to invite the audience with questions or comments, and especially those of you from ILSI Europe who may have worked on the BRAFO project over the many years. I've put some questions together and bounced them off the panel. They've seen them, but have not had a lot of time to look at them. And going back over my almost 35 years of dealing with caffeine and coffee and health issues, first question, are there really just too many disease endpoints that we have to deal with? Does it make it impossible to even think about doing a benefit risk analysis? Is it too complex? People have heard me say in all of these years, it's been a privilege to be involved in this because you almost get to study every known human disease and every animal toxicity when it comes to the study of caffeine. In that problem formulation step you saw in the BRAFO schematic, you look at individual risks and individual benefits, and you look at the endpoints and see if there's endpoints that you could carry forward, first qualitatively, and then trying to do a more quantitative analysis as you saw it presented in the third and fourth tiers. A question, do we really have benefits that are well enough established through the weight of the scientific evidence to even continue the process, or in our case, to actually start the process on caffeine? Do we have the quantitative necessary data necessary to do this benefit risk analysis? The metrics that BRAFO and others have used in doing benefit risk are DALYs and QALYs, the disability adjusted life years and the quality adjusted life years. I have no familiarity with that literature, but those are the metrics that are used. We've heard today, we've talked about pregnant women as a sensitive subpopulation, children as a sensitive subpopulation. In our last presenter, Dr. El-Sahimi, talked about the sensitive adult human population that may have the slow and fast acetylation going on. So how would we handle these sensitive subpopulations if we were going to try to do a benefit risk analysis of caffeine consumption? And the final question would be, you've heard that we've got caffeine in naturally occurring sources, we have added caffeine in products, and would you have to handle those differently in the process? So let me leave these questions up here, and I'd like to kick it off by inviting any of you panel members who would like to jump into any of these questions, perhaps even question number one there, the complexity of the endpoints. Is there just too much to take on to do this balancing of risks and benefits? And I'm looking for a volunteer. There he is.
So I also want to disclose, like you, Jim, that I am also a friend of caffeine, despite what you may think from the results. But personally, a friend of less than 200 milligrams for myself, because I know I'm a slow metabolizer. The focus of my presentation was on cardiovascular disease, but you mentioned other health outcomes, and of course we all know that caffeine specifically has been linked to not only some adverse effects, like I mentioned, cardiovascular ones, but a beneficial effect on things like Parkinson's, liver disease, certain types of cancer. In fact, we did a study with Steve Narod, who is one of the co-discoverers of BRCA1 and BRCA2, who had observed that coffee was associated with a lower risk of breast cancer in women who are carriers. And because there have been some carcinogenesis studies showing that caffeine can inhibit tumor development, we thought, well, let's look at fast and slow metabolizers. And as one would predict, the protective effect was due entirely to the slow metabolizers, because the fast metabolizers get rid of it very efficiently. So caffeine didn't have an opportunity to have a protective effect in those women. So again, it's not about pointing out that, where it's causing harm and where it's causing benefit, but it's using genetics as an additional tool to improve measuring exposure internally. So again, when we look at all these different health outcomes, I think it's important to kind of revisit that data to see whether or not that type of internal stratification could give us a clear view of what caffeine might be doing in those different diseases.
As I put on the slide there, the discussion we've heard this morning about sensitive subpopulations, this is the existence of the polymorphisms for the general population. Is that going to prevent us from doing what we're even talking about? I mean, nobody knows, or not many people know whether they're a fast or slow metabolizer. Can we even talk about some of the other balancing of risks and benefits?
Well,
in the absence of knowing whether you're a slow or fast metabolizer?
I think, first of all, the research is lagging in this area. So before we can even look at what we tell consumers, we have to provide the data. And so other than the few examples that I've given, I think, that as you mentioned earlier, there's tons of studies that have looked at the effects of caffeine on every health outcome. So we have to go back to that data, I think, and look to see if we can identify genetic subtypes. CYP1A2 is just one nice target because it happens to be play a major role in the metabolism of caffeine, but there could be other genes that code for proteins that are the targets of caffeine in different tissues that could explain other effects. So I think we need to advance the science in this field before we can look at how we can use this information for recommendation for doing a risk-benefit analysis.
Comments from other panel members on this question of the diversity of the effects?
Yeah,
whether it's possible or impossible to even go forward?
I don't want to take on possible or impossible, but I do want to note that people, the bulk of our population, are freely able to refuse or consume caffeine. It's a personal choice, which we allow people to make, and we allow them to consume caffeine in a variety of different forms. The benefits of the selections that they make are, we believe, from the point of view of psychology, perceptible and desirable. That is, you know that you feel better day in and day out when you have your coffee versus when you don't have your coffee, and data on measurements of psychological performance confirm that there are not only benefits with regard to how you're feeling, but your ability to perform, including performing tasks which involve risk. So you can track back, for example, to number of car accidents and get a number in theory that suggests caffeine has protective effects on individuals who are driving when they're tired, for example. But traditionally, those kinds of benefits aren't integrated into risk-benefit analysis because the risk-benefit analyses focus more on medical diseases. And not just driving, but feeling better is also a benefit which is real and important, but one that is difficult to, it's hard to develop an algorithm that would weigh that as a benefit when it actually is a fundamental reason that we're consuming the product.
Andy, you have some interest as well there.
I'd like to just go back to the point that my colleague on my left made about needs to go back to the science. I agree completely, and I think what you need to do is understand the underlying mechanisms. And if you think about how that relates, for example, to disease outcomes, you might want to address the issue of perhaps caffeine in the immune system, which is going to have enormous implications for a wide variety of conditions, rather than necessarily looking at all those conditions individually. And on the positive side, one of the big advantages of a lot of the behavioral work is we not only know what those effects are, but we know the plausible underlying brain mechanisms. And if you contrast that, for example, to a purely epidemiological approach, which is going in and looking for associations without necessarily having the underlying mechanisms, then I know which is more powerful.
We both of you, Harris and Andy, talked about the benefits of caffeine from the cognitive and the physical performance side of things going back, this goes back 30 or 40 years. But Harris, I mean, we're not talking disease endpoints here, so are the cognitive and behavioral and performance effects off the table in the, I mean, in the process you identify the benefit you're going to compare to the risks. Because it isn't disease, is
Right.
it
Well, I would argue that it should be on the table
on the table?
because it is a real benefit, and it's a benefit which people are able to freely choose. So, and it's ubiquitous when 80% of the population engages in a behavior. That's a lot of votes for having the opportunity to consume that beverage or whatever form it's consumed in, and use it as a daily part of your life. It's truly a lifestyle of choice, and I don't think at this point we necessarily have good mathematical procedures to adequately weight those kinds of huge general benefits compared to one case of disease or X% probability of getting a disease.
I think as well, the whole approach is moving away from a medical disease-based model to think much more in terms of softer concepts like quality of life, well-being, and these are things which can be readily put into cost-benefit models.
Yeah, a comment and maybe a question to Alessandro. You didn't pick caffeine. The series of things that were looked at in the BRAFO projects, oily fish, the discussion related to the positive health benefits of fish consumption versus the methylmercury content. Soy was looked at, folic acid fortification of flour, replacing saturated fatty acids with carbohydrates. So the things that you've used in your case studies was kind of like just two things. And, there's contaminants in fish, but fish is good for you. And what I think we're going to be struggling with here is we've got so many endpoints. Did you think of caffeine as a possible case study?
But actually, no, not at the time. Alessandro from ELSI Europe. At the time, no. So was entirely on expert judgment on the kind of case study that we are able to identify. Also because within the remit of a European project, you should follow like a description work, so we cannot extend the kind of examples that we have identified there. But then the BRAFO framework could allow you to solve some specific questions. So if you would like to answer the question, caffeine is friend of foe, BRAFO cannot help you at all. What can do is, for example, if I consume 200 grams of caffeine per day in a particular kind of population, what is the risk and benefit associated with this? And then if you have the data and you have less uncertainties related to this, you can arrive to a quantitative evaluation of your question. And then we choose, the novelty of BRAFO was to have a common matrix to identify risk and benefit. Because historically, we are more keen and prone to have a more quantitation of the risk because we like to prevent ourselves to getting some kind of disease. But then we don't have the same quantitation for a benefit. So what is a benefit? Because most of the time you express the benefit as a risk reduction, but then it's not a real benefit, so you don't have the metric to measure this. So coming back to BRAFO and then your question, probably you need a little bit more quantitative data if you can. And then if it's not possible, it's entirely on expert judgment. If you can arrive to a qualitative evaluation that is robust enough to answer your question.
Thank you.
Hi.
I can't see.
Sorry. ELISA Gomez from Mexico. I think it's really interesting, and there is a lot of things to do because, you know, I work in a gastroenterology department as a nutritionist, and I have both sides of the point of view of the gastroenterologist, because in liver diseases there are a lot of evidence, and we have a trial with coffee, and after 2 cups of coffee every day, the people with fatty liver disease progress slowly than the people who doesn't drink coffee. And that's interesting because we don't know if it's coffee or if it's caffeine or it's another thing inside of it. And also there are some evidence about hepatocarcinoma, that people who drink coffee have lower risk that hepatocarcinoma when they have liver diseases. But we also find this in chocolate, so we don't know if it's caffeine, coffee, or theobromine or other things that are inside of it. But also, the gastroenterologist, a lot of people, of physicians take away coffee because they say it's not good for gastritis and that kind of diseases. So I think it's important to see every disease as different and also see every source of caffeine as different. That's my point of view.
Thank you. Before we go too far into the audience, is there any, let me just ask a question of Jennifer. We had a sidebar conversation about this sensitive subpopulation called pregnant women. I said, but you're not talking about the benefits of coffee consumption or caffeine consumption during that same period. Would you expand on that a little bit?
There we go. In terms of a risk-benefit analysis, it's challenging, I think, to conceptualize benefits to the fetus. You know, in my own interpretation of the literature, in terms of benefits to the mother, I believe there are a couple of studies that have suggested a protective effect with regard to gestational diabetes or insulin resistance, potentially. Although I haven't critically evaluated these particular studies, but the suggestion, the hypothesis is generated. So, you know, in reading through or taking a look, the BRAFO criteria, it's new to me, but in terms of looking at the recommended procedures, basically, you know, does it even qualify if it's not balanced, if the risks are not balanced with potential benefits? Would it even qualify as being an outcome incorporated into the question?
Right. Yeah. So separate handling of pregnant women.
A separate issue, perhaps, with a separate approach. You know, I have to admit I'm not accustomed to considering the benefits of adverse, of risky exposures.
Go ahead.
Gerhard Reckemer, Max Rubner Institute, Germany. I just wanted to inform the audience here that EFSA has put out a 112-page scientific opinion on caffeine on the 15th of January. And that already has created a lot of media attention in Germany because the director of EFSA have made the statement that if you consume more than 400 milligrams caffeine a day, it will be dangerous to your health. And he's made that statement public. So in Germany, this right at this time on the main TV news show, there will be a statement about caffeine and the dangers of caffeine to your health. And there will be, right after that, a great report in a political magazine on TV at prime time in Germany. Just for informing you, I haven't read all that 112 pages yet, but it's an important document, I think. And one of the members of the NDA panel was also a member of Bravo project, and what you have shown here as the results which are published from the Bravo experience.
Yeah, not many of us have gotten very far into the 112 pages of the EFSA report, the draft opinion that came out last Thursday. There's a 2-month comment period, and there's not a lot about benefit in there. It's about, looking at the potential safety levels for the general population and pregnant women. Brent?
All right, Brent Koblish from General Mills. I by no means am a caffeine expert, but sitting through this session all morning, I've seen some statistics, and I think that a lot of them were presented from a U.S. standpoint. 80 to 90% of people are drinking caffeine or consuming caffeine daily. 60 to 70% of pregnant women are consuming caffeine daily. And my question is, from a scientific standpoint, I don't see a problem. The risk assessment, from a risk assessment standpoint, it has to be population-based. Where's the problem from a scientific? Now, I understand the regulatory and the consumer perception, stuff like that, okay? But from a scientific perspective, I don't see a problem. Am I missing something? I guess I just don't understand why this has become such a huge issue, except from a consumer perception. But from a scientific standpoint, it seems pretty clear to me. Can someone fill
Anyone
me
on the-
in?
Well, I think we don't know if something is a problem until we look to see if it's a problem, right? Oh, sure, and they've also been consuming, processed carbohydrates, saturated fats. It was a time when we... Sure. Sure, and one person's, dose is a poison for one person and not someone else. But I don't think looking at the current prevalence of consumption can justify the safety of something. Absolutely not. You have to look at the evidence, and that's what I think we're trying to do here. And the way to look at the evidence is to look at the different associations. One thing that is clear is that caffeine is not universally harmful for either everyone or for all diseases. So it does look to be associated with a protective effect for certain conditions like liver disease, Parkinson's, others, but have an increased effect with other health outcomes. So we still have to look at the evidence critically. We have to look at the tools that we use to make the studies, whether it's observational or doing more RCTs. But I don't think we can look at the current exposure at all and say that, oh, it's safe, people are consuming it, they're not dropping like flies. People are dying, everyone is dying. So the question is what they're consuming and the amount that they're consuming contributing to that premature mortality? We have to look at that in an empirical manner. I agree 100%, but that's contributing to the evidence base. It will be ethically difficult to propose to do an RCT on caffeine and look to see, who develops a heart attack or who develops hypertension. But we have to look at the totality of evidence. So I tried to give an example where using genetics has now improved a little bit our understanding of the link between coffee and hypertension and cardiovascular disease. We see that people respond differently. I think we need to take the similar approach with other health outcomes. I didn't present data that we'll be publishing soon looking at kidney disease, but we see a very similar effect as well, where slow metabolizers seem to be at higher risk, fast metabolizers seem to be protective. So it's about looking at the data and elevating the quality of the science that we're doing and not just say, everyone's consuming it, let's forget about it, and it's safe.
Elane Kroll, DuPont Nutrition and Health. I think this whole session is great because it's a case study of looking at a specific nutrient or compound, I don't know if you want to call it a nutrient, in nutrition, and the way we should look at a lot of studies. And I particularly like the last talk because it does sort of address some of the issues and concerns that have been addressed in studies in general. And when one of the questions you posed is, do we have too many disease endpoints, to even consider to look at a benefit risk analysis? And I think we do have, in every study, we do have individual data, the ability to generate those up arrow, negative, no change and reduced change. If we did do, and my colleagues laugh at me because I'm always talking, let's do a waterfall plot where you actually see the hyper responders, the hypo responders, and then the non-responders. And you take any disease endpoint that you measure in a prospective trial, and you generate one of those plots. If you see great extremes at one end and non-responders in the middle, then you know there's something about the population that you chose where there's people that are hyper or hypo responders, that there must be a genetic contribution to that effect. If it's very flat and represents the mean value, and the mean is pretty much reflective of everyone, then it's harder to decide whether or not there's some genetic predisposition. But I think to what you were asking is that we will only be able to make these decisions, not do more studies and just take the mean value plus or minus standard deviation, say, oh, there's no effect. But if you take the studies, for instance, in the small gestational age, and look at that on an individual basis, is there something that we can dissect out of those, if they're prospective studies, that we can say, oh, the women that had small gestational age, there is a clustering of them. But if they all, dance around slightly around the mean, then it's harder to really be able to pull that apart. But it's more of a comment, but I think we need to analyze the existing data because practitioners are more interested in, when physicians or nutritionists deal with their subjects or patients, they want to give personalized instructions, and we do that all the time. And they get all the information that they can for their particular subject. That's why no two hypertensive patients are on the same medications. We're doing that all the time. So let's just analyze the data a little bit more deeply. Thanks.
I think one of the advantages we had that some of the RUFO experts struggled with when the risks were only on the animal side and the benefits were on the human side. Hadiya, is that true? A bigger problem. Here we've got wealth, years, decades of data on human studies on these endpoints, and we're not dependent upon the animal study. Sure, they helped us with mechanism and other things, but we've got human studies we can balance, and that was something that you guys struggled with in the BRAFO project when there was only the uncertainties that come in related to animal studies. We aren't dealing with that, thankfully. Question?
Hi. My name's Tom Davis. I'm the Chief Scientific Officer of Monster, but I'm also a professor of pharmacology, neuroscience, physiology for 35 years at the University of Arizona Medical School. So thanks for coming to Phoenix. My question is really about, I think it's a very complicated area, obviously, but pointing at SNPs and realizing that polymorphism can be a contributing factor to this is very real. I agree with that. But you have to weigh the fact, in the population, let's just take North America, for instance, you have to look at the rate of SNPs. You have to look at how many SNPs are we looking at? Are we looking at a single polymorphism with a cytochrome P450 enzyme? Or are we looking at a single polymorphism with catechol methyltransferase? Or are we looking at a population that is quite unique in their polymorphisms, of which we don't know how many there are. And there may be a lot of polymorphisms that are contributing to your event. And to say that it's just the caffeine molecule acting because a SNP at the cytochrome P450 enzyme increases its half-life dramatically, then to turn around and say it's the same with catechol-O-methyltransferase affecting cardiac events, I'm sorry, but I've been teaching pharmacology for 35 years. I have 40 postdocs, 30 graduate students. I would send them back and I'd say, guys, don't focus on the single event. Be aware of the fact that there's a lot of SNPs possible in that population that you're not aware of yet. And those may be contributing to the event that you're predicting to be a cardiac event or a contributor to hypertension or some other area. I appreciate the mechanism very much, but I worry sometimes that we focus too closely on an event in order to make a point in a given publication. And I'm worried about that part of it, number one. So I'd love you to comment on that. But the real bottom line is, folks, this is a molecule that's been around hundreds of years. There is more research on this molecule than any I have ever seen. Fortunately for me, I'm a blood-brain barrier drug delivery guy to the brain. I look at what caffeine does in that realm, and that's it. But the research in animals and humans has got us to the point where we are today. And I respect everything that's presented today. But at the end of the day, I've got to agree with the guy from General Mills. What's the deal here? I don't get it.
Let him. Ahmed?
So I think it's fine to say that there have been, hundreds of studies or more that have been done, but the question is, have the right studies been done? If we look at the dozens of studies that have looked at the effects on cardiovascular disease, as I showed, some show increased effects, some show no effects, some show decreased effect. And now there's at least a couple of examples from independent research groups showing that at least one factor, doesn't mean it's the only genetic factor, but at least one factor can help explain some of these inconsistencies. I did mention that as future directions, we're interested in looking at kind of pathways of genetic profiles that give us, enable us to further stratify the risk, because as the previous question was, we can't just look at kind of means and a standard deviation. If we plot each individual response, we will see some people at each extreme end and then everything in between. So how can we better predict who are the ones at each of these extremes? And while one SNP will get us a little bit apart, certainly there might be others that can give us better prediction.
And there's real good evidence of that in the cancer literature. Excellent literature on the SNPs and how many there are in a given population and the ethnic contribution to SNPs. It's in the literature. I've read it and I understand it. But I really don't know the answer to this. What is the percent in a North American population of the cytochrome P450 SNP, the CYP1A2?
Roughly 50%.
And what?
Roughly 50%.
50% of the North American population are slow metabolizers.
That's correct.
That's fascinating from the standpoint of the consequence to the coffee and the caffeine consumption.
We are quickly running out of time, and here comes Craig Llewellyn. Clegg, you coming to the microphone for the last question to the panel?
Thanks a lot. I really appreciate the panel's discussion here. It is a very complicated and difficult topic to address, and a very complicated and difficult physiological and biochemical pathways to look at this in this way. Just building off some of the discussion that we've had and making sure that we understand the complexity of the issue and how the totality of the evidence can really lead us in a direction of around what is risk and what is not risk, what is hazard and what is just supposition. Understanding that, as I think Tom Davis was going, is that there's a multitude of other pathways that play importance here. That's been acknowledged. There's also another aspect of that in terms of the large variation of substrates of the CYP1A2 enzyme pathway. Exogenous, endogenous, and how those relate to other disease states. So again, focusing in these areas answers some important questions. However, taking that leap from here to there, a very dangerous thing in terms of risk. So I think that the important thing is to remember that we can't make that leap. We can take, as I said, the totality of the evidence, looking at what the evidence has, what we have, what we don't have, putting it together and coming to some answers that can help us from a public health perspective. So any comments around those understandings would be great from the committee.
We're mostly out of time.
Just a
but
quick
go-
one.
but let's
I'm not
finish.
advocating any huge leap. I'm advocating baby steps. And I think by using some genetic markers, we can take at least a baby step forward in better understanding, moving away from just the current one-size-fits-all, which is 400 milligrams in many jurisdictions, to a little bit of a baby step and separating. But then we still have to delve deeper into the science to take further steps.